My name is Beth Cimini. I'm a senior group leader at the imaging platform at the Bird Institute, and I teach life scientists not to be scared of image analysis. I know a lot of the folks in this group, at least historically, and some of the other presentations I've heard have been more in the genetics and genomics space, so it's really fun and exciting to bring some of the perspectives of the kinds of work I do into this group. And I want to just start from a really basic idea, which is that there's a lot of cool computational biology that is out there that we certainly hope going forward really cool work can be done in this area. But from a strictly point of view of who is out there that can do this sort of work, there's a lot more biologists and folks in the biomedical sciences than there are mathematicians and computer scientists. And so from a point of view of being able to scale up, there's a lot more unmet need, or there's a lot more people who can fill these unmet needs in biology. Particularly when you start looking at women and other minoritized groups as well, though, you can see that there could also be an equity thing framed around this, where if we want to open up computational biology to people who have not had an opportunity to it before, it's probably pretty likely that those people are going to be coming from the biological and biomedical sciences. So I think there's no more time that is more important than now to really create tools where we give access to biologists in order to do good computational biology. And the field of image analysis hasn't really been traditionally thought of in this computational biology way. And for a lot of folks, that's just because that is not how they were trained. Microscopy analysis used to be about drawing what you could see, and hopefully you were good at drawing, as Ramonica Hall was. I myself am not, and would have been terrible in this era of science. But image analysis has typically been a pretty manual process until pretty recently. Just to walk you a little bit through my background, not because I think that my background is so interesting, but because I think it's probably pretty representative of the way a lot of people were trained. I fell in love with microscopy as an undergrad at Boston University. I was looking for research projects as a rising junior, and when I saw my first microscopy image and talking to a research professor, I fell in love. Figured if I was going to be spending hours a day and hours a week making data, I might as well be data that was pretty and nice to look at. And so I spent a couple of years working to create pictures like this that you can see. We have a cell that is producing a certain kind of neurotransmitter in green, and a signaling molecule that's downstream of neurotransmission in pink. We wanted to show, are these cells activating the signal, and are they doing it under certain conditions if we block certain receptors? But when I was pulling the paper together and asked, what should we do? It should be just representative image shown. And the advice I was given in terms of making sure that my image was representative was to print out all of my pictures on paper, put them all on the floor, organized by treatment, and then look at everything side by side to see what conclusions you can draw. So this was the way that we quantified our phenomenon. Thankfully, there was a grad student in the lab who was able to teach me a little bit of ImageJ, so I could do a line profile. So here we're drawing across these green and pink and plotting the intensity values, and we can see that the green and the pink, green is up above, pink below, do actually overlap to some degree, but everything was about just finding the most representative image of a phenomenon. I really liked biochemistry and molecular biology in undergrad, so I went on to do a PhD in it at UC San Francisco, and there I went to study telomeres. And telomeres are the caps on the ends of your chromosomes. In this image here, we have nuclei are in blue, the telomeres themselves are stained in red, and DNA damage markers are spotted in green. And I was really interested in the mechanisms of what happens during DNA damage at telomeres, what triggers it, what sorts of things are recruited to it, things along those lines. And again, once I started making these microscopy images and I looked at what was standard in the field and what people in my lab advised me in terms of how to analyze those images, what I was told was just count how many localized spots you have and how many non-localized spots you have, just print them out and, write down on each one exactly how many you have, and then sum all that up, and there's your data. But what I was really interested in knowing was what were the properties of the telomeres that had these certain things. Were they longer? Were they shorter? Were certain proteins there or not? And in what quantities were certain proteins there? And a non-cancerous human cell has 90-something telomeres. Cancerous ones tend to have a lot more. And I wanted to look at this for lots of different treatments and across hundreds of cells in each treatment. I wasn't going to count all that by hand. And I couldn't find out some of that information, like telomere length, in any sort of quantitative way at that scale, even if I did want to count it by hand. But no one that I talked to, even people who'd been using microscopes for a long time and were really excellent microscopists, was able to give me good advice about how to get started. And so this launched me down a career of being really passionate about image analysis and making it easier to access, because I was incredibly frustrated during a lot of these periods and had a really difficult time doing the research that I wanted to do. And so when an opportunity for a postdoctoral position at the imaging platform at the Broad Institute opened up, where I could help make these sorts of tools and help teach people how to do these sorts of things, which I was already doing for free as a graduate student, the chance to actually make that my career was incredibly, incredibly valuable. And I've been there ever since, and they'll never get rid of me. So the imaging platform is a really fantastic place. They do a lot of really interesting work about making microscopy analysis more high content specifically, in addition to easier to do. I'm really not going to talk about any of that in the interest of time, but we're really interested in making images a first-class data source where we have lots of features. In a typical what we call cell painting assay nowadays, we extract about 6,000 features from each cell. And once you have these really high dimensional data sets, much as you can in something like a single cell RNA-seq, we can start clustering features, clustering cells with similar features, and looking at relationships between clusters to do biological discovery. So you should check out the other things the platform does that I'm not going to go into today to learn more about that. The tool that I eventually found in graduate school and that I continue to work on to this day is called Cell Profiler. And it's been around since Ann Carpenter and Ray Jones put it together in the mid-aughts. And so it's more than 15 years old at this point, which for an open source software package is pretty high longevity. One of the things that I think makes Cell Profiler really special is the people who make it, and is in fact really the only reason I show this slide. And I've color-coded us in a couple of different colors because I think one thing that adds a lot of value is that there's always been at least one biologist who here I've color-coded us in green, heavily involved in the creation of the code. We've had a number of excellent professional software engineers over the years who have, of course, been critical to Cell Profiler's success. But having someone who comes from an end user perspective, knows how biologists think, the kinds of analysis they want to do, the kind of terms they use in particular situations, I think is really what's helped Cell Profiler stand the test of time. So we also have a companion program that we call Cell Profiler Analyst that I'll also show you briefly during this talk. But so there are two parts to image analysis that we really think about. Cell Profiler is for image analysis and quantification. It is for finding the things that you care about and measuring them. And we hope measuring them in lots of ways so that you can do things like morphological profiling. Cell Profiler Analyst is for once you have the data out, how do you then explore it? How do you get the answer to a question? So we sometimes refer to these tools as measure everything and ask questions later. So it's really important to us as we're making Cell Profilers to make sure that we're actually responding to the needs of the community and what our end users want, what they need, what niches are already filled, and where can we add particular value. And so in very fortuitous timing, the pretty version of this went up online earlier today. A bioimage analysis survey that we and some of our partners through the Center for Open Bioimage Analysis conducted in late 2020, where we talked to about 500 different people about what image analysis needs were, things about their demographics. I'll go into a little bit more detail. One thing we did end up recommending in this paper, though, is that it's going to be really critical to create more resources to train the trainers. And so we singled out life science trainers as one of the really great initiatives that we're aware of. And so we're hoping that this can be expanded and others will come into existence. So one of the sets of questions that we thought was really critical in figuring out who our users are and what they need, especially so that we could see if different kinds of users had different kinds of needs, was asking them to describe their work, whether they were mostly spending it in the wet lab doing nearly entirely imaging, creating samples, acquiring images on a microscope, or doing nearly entirely image analysis, someone like me who's in a dry lab now and spends all of their time analyzing images somebody else took, or somewhere along that spectrum. We also wanted to know how people rated their computational skills and their computational comfort in developing new skills. We had a strong suspicion that those would be related, but perhaps not exactly the same. And so here's just one figure from that paper. And what you can see is that, first of all, we have people grouped by how they would describe their work on the x-axis here, and then we have them color-coded by their self-reported computational skill. One thing that jumped out to us immediately is that almost certainly our survey is not representative. Perhaps unsurprisingly, people who take a survey about open source image analysis tools are a lot more likely to be users of open source image analysis tools, because only about 10-ish% total of the people in our survey described themselves as spending most of their time doing sample prep and doing microscopy. And from our experiences, that's just not representative of biology as a whole. But it does mean that this is the group that we really want to focus on, because the ones who describe themselves as spending at least half of their time doing image analysis at least probably know how to get started doing these sorts of tools. The people in what we call the imaging group who rated their work as 1 or 2 on the spectrum of microscopy to analysis are probably who we need to spend the most time designing content for. If you look carefully at the different colors here, one thing you might notice is that the top level of computational skill is entirely absent in this group. So no one who's reported that their work is in this group felt that they had excellent computational skills. And the mean of this group is the lowest mean of any of these 3 work categories. And the mode is only 2. So we know that we have some really computationally self-reported unskilled folks in this group. And when we looked at the relationship between the skills that people already had and their comfort in developing new skills, again, we suspected that these would be pretty strongly correlated, and indeed we see that they are. We weren't sure what effect someone being a trainee, that is an undergrad, grad student, or postdoc would have. Didn't seem like it changed things terribly much, although the ones who said that they didn't start out with very many computational skills were more likely to be comfortable than folks who said that they weren't trainees. But in general, the people who report low computational skills, which are overrepresented in our imaging group, aren't necessarily terribly comfortable in developing new computational skills. So any approaches we develop, we really need to keep that user group in mind. Again, one other thing that made us pretty sure that our data is not totally representative, most of the folks in the survey reported that they are using open source image analysis tools. Again, not that surprising considering who wrote it and distributed it. But we also see that folks who are in these lower image analysis work groups, so our imaging group, our 1s and 2s, are really not using computational libraries and scripts very much, which we expect based on their self-reported computational skills. And they're basically almost always using either open source point and click software, or maybe some, if they have access to a commercial product, something like Amaris, using that. But they're pretty much left out of these computational libraries and scripts that are where a lot of the really exciting image analysis developments are happening. So in terms of how we actually usually try to reach these people, we're not at a degree granting institution, so we don't end up having a ton of contact with undergrads and grad students. So the main way that we do this is in 3 to 5 hour workshops. Since the beginning of 2010 through March 2020, we ran 132 workshops, so it's about 1 a month, 20 to 40 people typically, so you can ballpark that at about 4,000 people. And like I said, these were typically 3 to 5 hours long, enough to sort of teach people the basics of how to use the tool. And then we had different ways to talk to people afterwards, which I'll go into towards the end of the talk. Since March 2020, obviously our strategy had to change. We have done some webinars, a little bit less than our previous 1 a month, and we've been getting, again, about the same size, about 40 people each. but we've really pivoted a lot to video tutorials. And a big reason for that was the data that we got back from this survey. What we see here, and this is grouped by computational skill level in the same way as before, we did a 1, 2, 3, 4, 5, 6, 7 categorization. And people who report that they're at the lowest computational skill level when asked how they want to learn particular things, really strongly responded that they would like video tutorials. They're fine with written tutorials as well. They're fine with a lot of other things, but absolutely the strongest response that we got was for video. Some of the things that might be more, say, academically incentivized, like best practices articles, which we do still write some of those, are not actually the right way to reach those users. And so anything that we want to put out, we need to be careful about format, because the difference between 71% of people being very interested versus 22 might end up creating some real disparities in terms of who we're actually able to reach. And we think that sort of the proof is in the pudding here, in that we've put together about 15 different video tutorials in that time period. And we have several that are over 1,000 views, and our most popular is about 20,000. And so in a period of a little bit less than 2 years, we've been able to reach 5-fold what we did in the 10 years previous. And we also have some of all of these tutorials that we've written tend to have written versions, and we have example pipelines on our website. So I think the best way for me to show you how we teach Cell Profiler is to do a little bit of teaching you Cell Profiler and showing you the program. And I'm going to do it in the context of the actual experiment and the actual workflow that we do in our most common introductory tutorial, which is our translocation tutorial. So here we are looking at cells. They have a DNA marker, which is in magenta here, and they have a GFP, which is green fluorescent protein, tagged transcription factor. And so this transcription factor under normal cell conditions hangs out in the cytoplasm, so it's not inside the nucleus. And then after translocation, when some environmental stimulus happens, it goes into the nucleus, starts co-localizing here, and goes to the DNA and turns on certain genes. And so this is real data from a real experiment where a dose response curve was done. This level, you can possibly guess from the fact that there seem to be a lot more cells on the left here than on the right. This level causes complete translocation, but is also pretty toxic. So one of the things that we teach folks is in about 3 to 5 hours, although we've done it in as little as 1 hour, can they get the actual IC50 of this drug and figure out how much drug you need to actually cause full translocation. So this is Cell Profiler. Cell Profiler is essentially an image analysis workflow tool. What we have here on the left is what we call the pipeline panel, and Cell Profiler's concept of a pipeline is that we will just stack image analysis operations in order and apply them to as many image sets as you have, whether that's 10 or 10 million. And because we save these in simple text files, you can reuse and reproduce your data, send it to somebody else, etc. So everything that Cell Profiler does is organized into these modules, which some of them are doing things like pre-processing an image. Some of them are about finding or manipulating objects, creating measurements, creating files, things along those lines. And so to give you a little bit of scale about what we're doing here, in terms of having something that is small and tailored and only accessible to a certain group of people versus something that is sort of huge and flexible, but therefore a little bit anarchic, Cell Profiler has definitely gone the latter route. We have 94 different modules, including the ones that are involved in importing data. And in those 94 different modules, we have 930 settings of a variety of different kinds of class. So there are a lot of different places where you can interact with Cell Profiler, and you can essentially therefore make infinite different kinds of analysis, which is a good and a bad thing. Within the modules themselves, there's 44,000 lines of code, but there's actually 20,500 lines of documentation and manuals. So we actually 30% of the lines that we've written here are actually documentation lines. The library as a whole is 23% documentation. It's lower outside the modules, but it's still pretty heavily documented. And because we have so many modules and so many settings, we find that that's really critical. So if I load in a pipeline here, see that a pipeline just consists of a number of different modules, some of which don't have very many settings, and some of them have lots. But every single one of those 930 settings has a question mark button next to it. And we think these question mark buttons are the most important thing in Self-Profiler. Anything that has 930 configurable parameters is going to be a little bit challenging to learn to use. I've been using Self-Profiler a very long time at this point, and there are still some things I'm like, oh, I forgot it did that, or I forgot that's how that worked. But we want people, as much as is possible with a program of this size, to not get lost, or at least to have some idea, to find out the information that they need. So particularly in the most commonly used modules with the most commonly used settings, we spend a lot of time writing documentation about what are good things to do, what are less great things to do, under what circumstances are things particularly appropriate or not. And again, I think a big part of the reason that this software has succeeded is because the folks writing a lot of this documentation have been folks from biological training backgrounds. And so I'm showing you here just about 4-ish months worth of commits from right before our Self-Profiler 3 release, which was the release where we added a major functionality, 3D support. And our 2 full-time professional software engineers, you can see their commits outlined here in blue, but we entirely rewrote the documentation at that point, and there was a tremendous effort from a lot of different people to look through every line of those 930 settings and make sure that we had the documentation that was right and made sense and was explained in a way that an end user could actually appreciate. And so I think that is what really helps people not get entirely overwhelmed. So what I have here, I've loaded up the final answer to the pipeline, but this is not actually typically what we would give a user when they sit down in a tutorial with us. What we actually give them is a relatively minimal pipeline, which then when they load in some data, we can open up some images. And it only has a single module in the pipeline outside of image input. We also give them a PDF with some instructions to follow. This PDF assumes that this person is not actually sitting in the room with us. It's designed so that if you wanted to do this entirely on your own, you could. But we do give them a set of written instructions on what steps to follow. I'm going to sneakily delete this for reasons I'm not going to get into. But when I go into Self-Profiler's test mode, which is the mode where you can configure different settings, one thing, whether or not you're an image analysis expert or not, you'll probably appreciate is that in this image, there's probably not only one nucleus. We have lots of different nuclei here, and they're not being found correctly. And it's not just that I loaded the wrong file or that my data doesn't match my file, it's that we've done this for a very particular reason. And it's that theory is not everyone's strongest point. And for particularly the users that we think probably need the most assistance from us, the second we start showing them a histogram and start talking about the thresholding, which is always the first step in a classical computer vision object segmentation, a bunch of those users are going to be gone the second this histogram goes up on the screen. It's just not something that they're comfortable with. And so this is an actual slide from the actual slide deck when we give this tutorial. And I think it's important to show users that mathematically this is what's true, and that there are certain methods that are more or less appropriate in certain situations. But the best way we have found that they can learn actually what the consequences of that is not to sort of connect the theory to the settings, but actually start playing with the settings and figure out how to fix something that's broken. It's the old adage of it's a lot easier to edit than to start from a blank sheet of paper. So one aspect that is wrong here in this pipeline that I can show you is that we have something here called a threshold correction factor, which is essentially just a number that is multiplied times the calculated threshold value. And so when we look at Cell Profiler's interface and see what is circled in pink versus not, we see that actually, surprisingly, our threshold is not actually too strict. It is not too far to the right, but it's actually too far to the left. And that's because we've chosen an inappropriate thresholding method based on the description that I just gave. And so we allow users to have the slides that walk through the theory right alongside the pipeline that's configured incorrectly so that we can encourage them to tweak all of these settings and to actually use that to see, oh, yes, I'm starting to get intuitive understanding of how this setting works. It's a lot easier for a lot of learning styles to then see the difference between setting this number at 1 and at 4, and then to think about conceptually the histogram and the thing sliding to the right versus the left. One thing that we also really like about this model is that at this point, we give users, in a typical about 4-hour workshop, we would give them about an hour at this point to fix everything that's wrong in this module and to go through and then set up the rest of the pipeline. And most of that time will be spent fixing up this one module that's wrong. On the one hand, one of the reasons it's helpful is, again, so that they can develop this intuitive understanding, but it also gives us as developers and as teachers a chance to actually watch them do it, to see what the pain points are, to see where people are continually confused. And I would say these tutorials that we give are one of the biggest drivers of major changes to our software. We entirely changed our build process in 2018 after we had done a number of tutorials where we found that telling folks they had to install a Java development kit from the internet, even if we provided a link, and then install Cell Profiler, we were still spending about half an hour of each workshop getting Cell Profiler fully installed on people's machines. So even though we had to spend a bunch of developer hours on getting that JDK packaged with Cell Profiler, it meant that we were saving an awful lot of end user hours. And we've majorly redesigned some modules, such as this Identify Primary Objects module, which now when you open it up, most of the settings are hidden in a way that's a little bit less scary and intimidating if you were adding this from scratch in a brand new pipeline. Because we spend these times when the users are trying to fix things or broken, watching them fix it so that we can improve documentation or things about the software or just things about the way that we teach it. One thing though, when folks are in this part of the process where they're really frustrated and they're trying to fix things, that we do think is critical to sort of getting them through that is by creating buy-in. Right here where things are broken and they're having a hard time fixing it is about the time when folks are typically like, well, why doesn't this just magically work? I can see where these nuclei are. There are all these little white circles. Why can't the computer do it? Or at least why can't I just outsource it to somebody who knows what they're doing? And I understand that it's extremely tempting. And so this is typically also a reason why we try and get buy-in from users by connecting them to the actual biology that they're studying and showing how their biological insights matter. And so these are just a couple of excerpts from one of our more popular blog posts about making image analysis pipelines, but we try and drive home to them as they're doing these tutorials and in communications with them later, that actually what is the right segmentation and what are the errors in segmentation that are permitted are going to depend based on your biology. A segmentation that is right in most images, but is right in all of the negative control images and none of the positive control images, is not actually something that computationally we can just say, oh, well, it's 95% accurate. We need that user input to say, what are the different classes there? And are my mistakes balanced across classes? Simultaneously, there are certain errors that, depending on the biology that you're studying, are pretty much okay. If you're trying to figure out if something is changing the mean intensity level of something, the exact outlines of the cell are probably not terribly important. But if you're trying to test something like size or polarization, they are really important. And so by showing the users that, no, this isn't just somebody's making me do computer science when I don't want to, but that actually the biology here is really critical for getting the right answer, we find it helps generate some buy-in and get them to realize that it's worth them spending their time, which nobody has enough of. Everybody has lots of other things they'd like to do, in actually learning to do this specifically. So I'm going to skip ahead now to the way we do that in the context of our tutorials, which is to then go into cell profiler analyst. So in the context of a typical tutorial, they would have spent about a couple of hours finishing this pipeline based on the instructions of the person in the room and by following the written tool. One thing we mentioned in the earlier call that is something that we've found has been improving things in some of the more recent, and by that I mean 2 or 3 tutorials that we've given, is by having one person assigned to this document, a second person in a group of 3 assigned to operating cell profiler, and a third person assigned to looking at the slides. And so by making them all work together, we can connect all of those pieces much more strictly, and we can have people see exactly how the slides are helping you figure out the workflow, and get folks just talking and excited. And it helps get people over some of the places where people get a little bit stuck. But they would be creating this pipeline, and then once they'd done it, they would run it on the 26 image sets that are provided, and then go into cell profiler analyst. So the major thing we have them do in cell profiler analyst is to then do a classification between our negative and our positive sets. I'm going to change, for the sake of anyone colorblind, change this to green and magenta instead of green and pink. And also I'm gonna make these guys, make these a little bit bigger so it's a little bit easier to see. Yeah. Okay, it's a little too much bigger, but that's about right. So one of the places where biology sort of starts to become apparent to them and they get pretty excited about is that when they get to this stage, usually a significant fraction of the students will notice that there's actually a third class of cells that we didn't tell them about. There's cells where this green fluorescent protein isn't expressed in the cells at all. And so having them say, oh no, I see that there's biology here and I can now make a classification decision about how I treat it. Do I decide that I don't classify those cells at all? Do I treat them in one class or another? And having them learn that, what they know about the biology and why cells might not have that protein, and having that be really critical to their decision, I think really helps create that sort of buy-in where they're like, oh, this can't just be done by a computer scientist who doesn't understand my problem. It needs to be done by me who does understand my problem, in partnership with computer scientists who are helping make it possible. And so this is a pretty easy visual phenotype. And so we'll have them train a classifier. In this case, it's random forest, but we provide a number of different ways of doing this. They can look to see if the features that come up make sense. One of the features that's coming up is the most important feature is the correlation inside the nucleus of the DNA and the GFP. So that's a feature that makes sense in terms of being predictive between here and here. And we can show that it has pretty good predictive value if we ask it to show us positive cells. It's made one mistake, but of the 20 that it showed me, I think 19 are probably right. Likewise, when I asked it to show me negative cells, I do have a few of these no GFP cells, but I don't have anything that I would overtly call positive. And I can also even tell it to show me cells that it's not certain about. And so when they see that in really just dragging and dropping a few things, they can start to create classifiers that work and have predictive value. This is when the smiles, when the ones that have gone away come back, and the students get really excited about this. I can then score all of the cells in my experiment. I don't need per cell data now. I can save this up to my database. And I can then have them generate at the end a biological measurement that makes sense to them, which is how much of this drug causes this phenotype. So if on the x-axis, I graph dose, and I'm going to do so with a log scale. And on the y-axis, if I grab my hit table now and I grab my enrichment of positive, which is my translocated cells, in about 4 hours, or here in about 10 minutes, I can actually make what looks like a pretty standard IC50 curve, and I can calculate the amount of drug that actually causes translocation without causing all of the cell death that we see. And so we are able to show students that with combining the computer science, which we've, as much as we can, explained to them, but made it so that they don't have to operate, and the biology, they can get answers really quickly. And so just to sort of wrap up here, one of the major points that we found have been, first of all, the more complex the task, the more the documentation you need. Cell Profiler is an incredibly complex program, which provides a lot of flexibility, therefore needs a lot of documentation. And by working with the domain experts on the documentation in partnership with the computer scientists, we found that that's where we get as close to right as we can, as close to useful for our end users as we can. If we give the students time to fix things that are broken and to edit, it helps them connect the theory of how different kinds of algorithms work to the practice of actually switching between algorithms and see what changes. And it also gives you as the instructor time to see where they're confused, fix what their pain points are, and where possible, do something different next time. And just show them that the biological knowledge that they have is critical to getting the right answer, and that they are a critical part of this doing good computational biology, and they bring a necessary skill to the table, which I think really helps provide confidence. We do also spend a lot of time thinking about, again, we're typically only interacting with people for a few hours at a time. How can we help them once they walk out of our tutorials? And we're now up to 25-ish thousand people served, but there's an awful lot more biologists than that in the world. One effort we've been really excited to be a part of is this forum.image.sc, which has emerged as sort of a nucleating point for open source image analysis tools. We're well over 40 official partners at this point. You can see many of them here. And we have over 140,000 posts and 27,000 threads. And the idea was to create a place that is tool agnostic, where someone can just upload a picture and say, hey, I have this picture, I have this question, where do I start? What tool is right? We've gone in 10-ish years in the image analysis space from having too few tools, or tools that maybe weren't powerful enough for a lot of use cases, to having so many tools, it's kind of daunting to figure out where to get started. So having a place where it's staffed by experts and a lot of different tools to sort of suggest, oh, mine's not the right one, you want his over next door or hers over that way, as well as incentives for the developers to keep hanging out there with things like conferences and job opportunities. So we think this is an important step, and we think it's been a really important part of helping nucleate this community, although there are certainly other great groups like New Bias, the Network of European Union Bioimage Analysts, who have done amazing work in this space. But we know there's still things that we can do better. The biggest thing is how do we reach people who don't know we exist? Creating a central place for them to try and find us helps, but for people who don't know that this resource is out there, then we're still not reaching them at all. And again, the fact that in our last survey, we only had about 10-ish% who described themselves as mostly using microscopy. Some of that will be those people just don't want to take a survey, but a lot of it is probably people didn't even know what was out there. How can we simplify entry into this really complex space? One thing we've heard anecdotally from a lot of users is that because we have made a space where technical discussion and low-level non-technical discussion are mixed together, that the fact that there's a lot of technical discussion going on, if you're somebody who is not really confident in your computational skills, actually makes it scarier, because you see these high-level discussions, be like, this isn't the right place for me. So do we actually need to think about different ways to present this data or different ways to manage these spaces so that users of any and all levels feel welcome? And how can we make it so that users actually don't need to know some of the things that I spent a lot of time telling you about? We certainly want users to understand algorithms and to not be sort of just throwing data at classifiers willy-nilly and drawing conclusions that aren't justified. But for things like finding an object within an image, there are a lot of tricks to doing it right that take a long time to learn. I can speak from experience on that. And so if we can make ways that need fewer configurations, which maybe shifting to things like deep learning, there's some great tools now for blobby type objects that have been designed for nuclei but work on sort of anything that's round. We are hoping that maybe by using those sorts of tools, we can make this space more approachable by being able to hide more of the sort of high-level computation from users if they don't really need to know it. We're also pretty optimistic about the promise of web tools. Installation has gotten easier and better with a lot of tools, but we still run across tools that people have put out where it doesn't work on Windows or on Mac, or it doesn't install at all. And so we're experimenting with the idea of creating more web tools to avoid installation so that we can get these tools to just anybody with a web browser. And so we're actively looking to just figure out what are the ways that we can take the things that we're already, we think, doing well and just put them in the hands of more people and make this a more welcoming and open space for everybody. And so with that, I want to thank the members of my lab, as well as our sister lab, the Carpenter Singh Lab, and Carpenter, who is the senior director of our platform, the original creator of Cell Profiler. And again, they're doing really great bioinformatic work there. The folks who funded us to work on tools like Cell Profiler and Cell Profiler Analyst, and the folks who've given us all of these beautiful images. We're a totally dry lab, and so all of these images have come from somebody else doing biology in the wet lab. And so thanks you guys for this. Happy to take questions.